Let's discuss a news from today's article, that is how gene therapy could cure cancer. First of all, let me explain briefly what the news is about. Scientists in United Kingdom have tested a new form of cancer therapy which reported success in a teenaged girl, Alicia, who has T-cell acute lymphoblastic leukemia. She was the first to receive this experimental gene therapy, which has relied on a new technology called base editing. It is done to fix our immune system in order to stop making cancerous T cells. The recent investigations have suggested no signs of it according to BBC. Now let's study the topic in more detail. What is T cell acute lymphoblastic leukemia? It is a form of blood cancer. Here the T cells, that is a class of white blood cells, will turn against the body and end up destroying healthy cells, which will affect the immunity. And this disease is rapid and progressive and is usually treated by chemotherapy and radiation therapy. So we use chemotherapy and radiation therapy for treating it usually. Now let's see why this experimental technique was done on her. It was done because she had done various standard treatments before, including chemotherapy and radiation therapy, but had limited success. So she had enrolled on this trial experimental medicine testing. Okay. Now, what is this technique called base editing? Here we know that a person's genetic code has 4 bases, that is adenine, guanine, cytosine and thymine, and they are having different permutations. Now these sequences can be used to produce wide array of proteins necessary for the body's function. Here T cells perhaps may have been misarranged in her case in the sequence of bases and had become cancerous. So biomedical engineering field has tried to put on a technique that allows genes to be altered or errors fixed. The most popular approach is CRISPR-Cas9 system. Okay. Now let's see what it is. It is an enzyme that acts like a molecular scissor. It is used to cut pieces of DNA at precise locations, and a guide RNA can be used to insert a changed genetic code at the sites of incision. Okay? This technique of base editing, even though it is a nascent technology, is more effective to treat blood disorders which are caused by so-called single point mutations, when a change in single base pair can cause terminal disease. Now let's see how this base editing is done. Healthy T cells were extracted from a donor and put through a series of edits. And finally, after spending a month in remission, she was given a second donor transplant to grow her immune system so that it could contain healthy T cells. Okay. Now finally, let's see how effective was the treatment. After 3 months of treatment, her cancer seemed to resurface, but most of the recent investigations suggest no sign of it. So that's all about the news. Thank you.